Very good morning, everyone. My name is Lakshmi Zadhav, and today I will be presenting my seminar on animal models for autoimmunity. This topic is from credit number 3, which is experimental immunology, under the heading of in vivo systems. In this presentation, we are first going to understand what is exactly autoimmunity, then what are the need for animal models and what are animal models, and finally, animal models used for autoimmune diseases. As we all know, our immune system is very finely tuned. It is running around the clock to prevent foreign particles, but at the same time, they are surrounded by self antigens. Our immune system does not mount and respond against our self antigens. This unresponsiveness of immune system towards specific tissues, cells, or substances is called as tolerance. When this tolerance is shown against our own cells and our own antigens, it is called as self-tolerance. How is this tolerance developed? What cells should be considered self and what cells should be considered as non-self or foreign? There is a simple criteria for that. All antigens present during an embryonic life are considered as self. So any antigen or any cell which is not present during an embryonic development is considered as non-self or foreign, and an immune response will be activated against those cells. So now our immune system knows that no attack should be mounted against self-antigen. Yet sometimes, due to many reasons, our tolerance mechanism fails and our immune system will attack our self-antigens. This is known as autoimmunity. Ehrlich in 1901 termed this as horror autotoxicus. Such diseases which are caused due to autoimmune responses are known as autoimmune diseases. These diseases can be either organ-specific or systemic. Organ-specific diseases are produced by targeting only one specific organ. For example, Hashimoto's thyroiditis. In this disease, only the thyroid gland is affected primarily. Next, diabetes type 1. Only pancreas will be targeted. Whereas in myasthenia gravis, only the neuromuscular cells will be targeted. On the other hand, some autoimmune diseases are systemic and affect the whole system instead of just one organ. Best example is SLE, systemic lupus erythematosus. In SLE, anti-nuclear antibodies are formed. These anti-nuclear antibodies react with nucleus of all cell types. Skin, kidney, joints, blood cells, blood vessels, all will be affected in SLE. I hope we are clear with what is tolerance and what is autoimmunity. Moving on to the main topic. What are animal models and what is the need for animal models? As I said earlier, we are talking about in vivo systems. In vivo systems involve studies done in living organisms. This study done in living organisms helps us to understand the reactions, the phenomena performed as a whole organism. But due to legal, ethical, and also religious reasons, we do not perform experiments on human beings. Hence, we need something which will provide us with similar results. Not same, but at least similar results are expected. Animal models are that something. We can perform experiments on animal models in order to understand the mechanisms of various phenomena. Genetically, most of the mammals are similar. Every mammal has developed its own complex and distinct function. Yet, all mammals share a significant amount of similarity. Also, most of the human diseases can also affect animal species. For example, diabetes, cancer, allergies, etc. Not only the diseases are common, but the infection mechanism of those diseases are common too. They are at least 90% similar. Researchers exploited this particular phenomena and used animal models to understand and obtain results about various diseases, their mechanisms, and also treatment strategies for the diseases in human beings. Thus, animal models are a non-human species which is used in research purposes. Natural, spontaneous, and induced. All types of pathological mechanisms can be studied in them. Most of the scientific breakthroughs were possible due to animal models. Millions of lives are saved every day thanks to all the testing and experiments done on animal models. Moving on to the main topic. What are animal models and what is the need for animal models? As I said earlier, we are talking about in vivo systems. In vivo systems involve studies done in living organisms. This study done in living organisms helps us to understand the reactions, the phenomena performed as a whole organism. But due to legal, ethical, and also religious reasons, we do not perform experiments on human beings. Hence, we need something which will provide us with similar results. Not same, but at least similar results are expected. Animal models are that something. We can perform experiments on animal models in order to understand the mechanisms of various phenomena. Genetically, most of the mammals are similar. Every mammal has developed its own complex and distinct function. Yet, all mammals share a significant amount of similarity. Also, most of the human diseases can also affect animal species. For example, diabetes, cancer, allergies, etc. Not only the diseases are common, but the infection mechanism of those diseases are common too. They are at least 90% similar. Researchers exploited this particular phenomena and used animal models to understand and obtain results about various diseases, their mechanisms, and also treatment strategies for the diseases in human beings. Thus, animal models are a non-human species which is used in research purposes. Natural, spontaneous, and induced. All types of pathological mechanisms can be studied in them. Most of the scientific breakthroughs were possible due to animal models. Millions of lives are saved every day thanks to all the testing and experiments done on animal models. Now, we know what animal models are and why do we need them. But there are many different animal species with many different strains within. In such cases, what animal should we use? Selection of appropriate animal model is very important. Some factors to be considered before selecting an animal model includes: first, relevance. It is of utmost importance that the phenomena which we want to study in the animal must be comparable to the humans. If we cannot compare humans and that animals in that particular phenomena, you should not select that animal. For example, if you want to study something related to limbs, that is hands and legs in humans, we cannot use zebrafish for obvious reasons. If we want to study an autoimmune disease, it must develop in the animal, either spontaneously or by induction. Relevance is the most important criteria for selection of animal model. Next is level of ease while handling. Animals smaller in size are easier to handle, and that's why most of the animal models are small in size. They can be easily held, experimented, and controlled. Also, large animals require larger finance, more maintenance, and more storage. Hence, they must be small enough to handle. But at the same time, they must be large enough to perform multiple biopsies at the same time. So size is of utmost importance. They shouldn't be too small nor too large. Next point is genetically well characterization. Well characterized animals will help you to give further insights about the experiment. Selecting genetically well characterized animals will definitely help. Fourth, rapid reproduction. Smaller animals generally have rapid reproduction cycle. Also, the number of offspring produced during every pregnancy are more than at least 2. This is very advantageous. We can obtain more animal models with lowest genetic variation. As Prajakta already explained, inbred training is done to reduce genetical differences. Lastly, lifespan. Animals should survive long enough to be usable. They shouldn't be diseased and must live at least till results are produced. All these factors play an important role in selecting an appropriate animal model. An ideal animal model may never exist, but it should at least serve as an example. Another example for relevance point on the top of my head is, if some disease affects only the primate species, then you have to use an animal model from the primate species. You have no other choice. That's why selection of a good animal model is very important. We have talked in general about all the animal models. We will now focus on models especially used in autoimmunity. Significant amount of immunological knowledge we possess right now has been developed using various animal models. They have given us an approximate idea about how a particular disease is caused and what countermeasures can be applied to curb that disease. Animals in general used are mice, rats, rabbits, guinea pigs, horses, and also primates if required. The most common and widely used, yet the most important model is mus musculus, that is, mouse. They have enabled us to create breakthroughs in immunology. This means if you predicted some genes in a mice, it's probable that those same genes must be present in humans too. So, why is mouse such a popular animal model? Because they're very small, hence they're very easy to handle and control. They have a very fast reproductive system and they also produce a lot of offsprings. And also, they have 90% gene similarity with humans. Also, its genomic sequence has been highly mapped. Its genome is well characterized. They can also develop spontaneous mutations. And if not spontaneous mutations, we can induce mutations into them. We can also use transgenic and knockout mice strains. All these reasons and many more reasons make mice the most preferable animal model for autoimmune diseases. In some inbred strains, autoimmunity can develop spontaneously, while in other, autoimmunity must be induced from outside. The animal models used for both these purposes are different. We will study them in brief. First, spontaneous animal models. Some animals spontaneously develop autoimmunity. In simple words, they develop an autoimmune disease which is somewhat similar to another human autoimmune disease. That's why such models can be directly used for experimentation. The results obtained from such animal models can be directly compared with humans. One of the spontaneous animal model is obese chicken or obese strain of chicken, OS. In this model, autoantibodies against thyroid antigens are produced. These autoantibodies slowly lead to destruction of the butterfly-shaped thyroid gland. This phenomena is very similar to Hashimoto's thyroiditis in humans. The autoantibodies formed first cause inflammation of thyroid. Because of inflammation, there is some leakage of excess thyroid hormone. This leads to hyperthyroidism. But after some time, because of the inflammation, the thyroid gland is unable to produce any hormone. Please notice we move from inflammation to hyperthyroidism to hypothyroidism. Some more similarities between humans and the thyroiditis present in chicken are presence of anti-thyroglobulin antibody. I repeat, presence of anti-thyroglobulin antibody. This thyroglobulin is a substrate. I repeat, presence of anti-thyroglobulin antibodies. This thyroglobulin is a substrate. Thyroglobulin is a substrate which is important for production of thyroid hormone. It also stores the inactive forms of thyroid. But once the disease is caused, anti-thyroglobulin antibodies are produced. These anti-thyroglobulin antibodies cause inflammation of the thyroid gland. Thus, OS chicken is used as an animal model to study Hashimoto's thyroiditis. The next spontaneous animal model is the NOD mouse, non-obese diabetic mouse. Type 1 diabetes is also called as insulin-dependent diabetes mellitus. So this non-obese diabetic mouse spontaneously develops a form of diabetes. This form of diabetes is very similar to type 1 diabetes of humans. Just like human type 1 diabetes, NOD mouse first develops insulitis. So what is insulitis? Infiltration of leukocytes into the pancreas. Infiltration of all the WBCs inside the pancreas. From these leukocytes, T cells are the most important. These T cells are overreactive and they do the most harm to pancreas. So once these leukocytes are infiltrated into the pancreas, they start destroying Langerhans B cells. We all know Langerhans B cells are responsible for production of insulin. Slowly, this infiltration gets very violent and later all the B cells are destroyed. And that's why very low or no insulin at all will be produced. Later, through experiments, it was found out that diabetes in a NOD mouse can be transferred to a non-diabetic recipient through the help of T cells. In order to understand the importance of T cells in IDDM, some experiments were performed using non-obese diabetic mouse. If you can see the left-hand side flowchart, in experiment number 1, a healthy NOD mice. What is the healthy NOD mice? It is a diabetic mice, but it is still healthy. So a healthy diabetic mice was exposed to X-rays. Due to exposure to X-rays, its immune system was destroyed. But to help the animal survive, bone marrow from a normal, non-diabetic mice was injected into the healthy NOD mice. After injection, the resultant mouse did not possess diabetes, thus proving after destruction of autoreactive T cells, diabetes will be diminished. To further prove this theory, autoreactive T cells from NOD were injected into a normal, non-diabetic mice. Diabetes was seen in such mice. These experiments in NOD mouse proved useful to understand the role of T cells in diabetes type 1 of humans. In these images, you can compare the amount of beta Langerhans cells in normal pancreas on the left hand side versus in diabetic pancreas on the right hand side. Thus, NOD mouse is a model animal for type 1 diabetes. A third spontaneous animal model is the New Zealand black mouse, also referred as NZB. This mouse spontaneously develops an autoimmune disease which is very similar to hemolytic anemia of humans. Autoimmune hemolytic anemia includes a group of disorders. It is not a single disease, it is a group of disorders due to malfunctioning in immune system. Due to the malfunctioning in immune system, different types of autoantibodies are produced which will attack the red blood cells. Due to the attack on red blood cells, it leads to hemolysis and also anemia. This NZB mouse can also produce autoantibodies against nucleoproteins, DNA, RBCs and also T lymphocytes, thus affecting the whole system. This disease affecting the whole system is very similar to SLE, as we talked about it earlier, systemic lupus erythematosus of humans. This NZB is considered as the best model organism for studying SLE. F1 hybrid is a hybrid which is formed due to NZB and NZW, that is New Zealand black mouse plus New Zealand white mouse. This F1 hybrid produces glomerulonephritis. Glomerulonephritis. That is inflammation of glomeruli in the kidneys. Also, granular deposits which are immunocomplex, they are deposited into the kidney, which may lead to kidney failure. So NZB mouse is also very important, but its F1 hybrid is also very important. Now, moving on towards autoimmune animal models. I hope we are clear with spontaneous animal models. So, but before we move on to autoimmune induced animal models, I have to ask you a question. So please listen properly and try to answer it. Okay, so Chidiac-Higashi syndrome is a genetic disorder. It is formed due to enlarged granules in leukocytes. If a human victim of this disease, if a human is affected by this disease, that human cannot survive above the age of 10. So you can understand the fatality of this disease. So now, interestingly, this same disease was found in a killer whale. I hope everyone is familiar with the killer whale. So this killer whale was kept in an aquarium in British Columbia. This killer whale developed the same disease. So a little background about killer whales. They are predators. They are also known as orcas. They are somewhat 30 feet in length and they weigh about 6,000 to 7,000 kgs in weight. So, if you have to cure that Chidiac-Higashi disease, you have to study it in an animal model. And you have an animal model, that is that killer whale. Will you use that killer whale as an animal model? That's my question to you. If anyone wants me to repeat it, I'll repeat it. Definitely no. We cannot use that killer whale as an animal model. Why? Because there are various reasons. Imagine the expenses, the amount of money required to keep a 30 feet fish in controlled conditions, the amount of people required to do experiments on that animal, the amount of more finances required to do the experiment underwater, because we have to keep that fish underwater to keep it alive. Also, you're going inside that fish territory. The swimming rate of the speed of swimming of that killer whale versus us is highly differentiable. We cannot swim like the killer whale. And most importantly, it has the word killer in its name. Why would you like to mess with an animal which has killer in its name? So no, you cannot use this killer whale as an animal model because it's very risky and financially it's very costly. So I was just trying to make you understand the importance of induction of diseases. Fortunately, we were able to induce that Chidiac-Higashi disease into smaller animals like cats and cattle. So we were fortunate about that. We didn't have to use that killer whale. So that's why induction of diseases is very important. So moving on towards animal models. The first one is the rabbit. So talking about rabbits. So in 1973, rabbits were induced with acetylcholine receptors from electric eel. I repeat, acetylcholine receptors from electric eel. Soon, the rabbits developed muscular weakness. I think everyone is familiar with the disease myasthenia gravis. In this disease, the ACH receptors on which acetylcholine molecule binds are blocked. Thus, no binding, and that's why no neurotransmission of the cell, which leads to weakness in the muscles. Just like in humans, those rabbits developed autoantibodies against acetylcholine receptors of eel. But these autoantibodies cross-reacted with rabbits' own receptors, thus leading to muscular weakness due to absence of neurotransmission. This animal model experiment proved that autoantibodies to the acetylcholine receptors were the cause of myasthenia gravis in humans. This was an immunological breakthrough. In this figure, you can see the skeletal muscles are loose or drooping in rabbits and humans with myasthenia gravis. Rabbit with myasthenia gravis is on the left hand side, but human with myasthenia gravis is on the right hand side. Our next animal model is experimental autoimmune encephalomyelitis or EAE. This model is used to study multiple sclerosis in humans. So what is multiple sclerosis? In multiple sclerosis, immune system attacks the protective covering of nerves. Nerves have a protective covering made up of myelin, and it is called as myelin sheath. Immune system attacks this myelin sheath, which leads to communication problems between brain and other parts of the body. This EAE model is the most and the best studied model in immunology. So this study can be induced in animals by injecting myelin basic protein or proteolipid antigen. Myelin basic protein is a protein which plays an important role in production of myelin sheath. This myelin basic protein is unique to the nervous system. This protein cannot be found anywhere in the body other than the nervous system. After a few weeks of injection, cellular infiltration starts. The myelin sheath is targeted by the immune system and that sheath is destroyed. This can lead to paralysis. In this encephalomyelitis, lesions are formed inside the CNS. These lesions are same in humans and the animal model. Mouse was used as an experimental model for EAE, and it helped us vastly to test treatment strategies available or treatment strategies which can be used against multiple sclerosis of humans. Next, a last induced immunity animal model is EAA, that is experimental autoimmune arthritis. We can experimentally induce arthritis into animals. This induction can be done using collagen type 2 molecules. These molecules start an immune response against themselves. This leads to inflammation in many joints. This EAE, that is experimental autoimmune arthritis, is very similar to rheumatoid arthritis of humans. In this slide, you can see the joints of a rat legs. A shows normal legs of rat. In B, we can see the swelling and redness from the posterior angle. Whereas in C, we can see the same swelling and redness from the anterior angle. Whereas in D, total deformity of the joint can be seen. So to summarize everything, animal models play a major role in understanding the mechanisms and causative agents of various human autoimmune diseases. I am done with my presentation. These are the references. Thank you. Please ask any doubts if you have.